So welcome to our hotliners. We designed this training especially for you because we want to forge a closer relationship between our plant diagnostic laboratory run by Katie Urria-Morwicki, who you'll hear from in a moment, and our fabulous hotliners, of which many of you are here on the video call. And I just wanted to share a few things that we have some more supports coming your way. So we not only have a new educational services co-coordinator who you may have read the email right before this. She is here and I'll have her say a few words. Judith Volkart, welcome, welcome. But we have a new staff member, Jonathan George, and his very first order of helping us out, he started this week, is to connect with the hotliners and see how we can better support you all with technology or processes or resources or whatever it may be. So he will be meeting with all the teams and the day leaders in the weeks to come and designing some trainings just like this one, so that everybody is well supported. And I just want to welcome Judith, who's a very exceptional person. I had the chance to get to know you a little bit. You were in the class of 2025, which is very unique for someone taking
Yes.
on our top leadership role. But congratulations for not only completing the course, but completing your 50 hours. And you come to us with a wealth of leadership experience, but I will allow you to share anything you would like with our hotliners, of which you will start working with in June when the baton is passed. And thank you so much to Karen Naff, who was really the utmost professional and did such a beautiful job and is leaving everything in a really well organized place. So Judith.
Yes, I'm very pleased to be here. Thank you so much. I feel like I found a home in Rhode Island after leaving California just 3 seasons ago to come here. I've met some of you. I've met the Monday team and I've met Cheryl and Susan from the Tuesday team, but the rest of you, hello. I will see you soon. I will be popping in just to say hello, shake your hand, see what kind of goodies you have there for us to munch on while you're doing your hotline. I'm very excited to be part of the leadership team here at the Master Gardener program in Rhode Island, and we are all so special to have the opportunity to be in Master Gardeners in this state. This morning I was perusing for a friend the Master Gardener program out of California, and it is not nearly as complete as the one here. It does not have a full hotline like we provide our home gardeners here in Rhode Island. And the number of workshops was very small compared to what we do here in our small state. So we might be small, but we're very mighty. And I'm very, very happy to be part of the Master Gardener program.
Beautiful. Well, thank you so much, Judith. And does anyone else have anything they would like to share before we get started? Right. So, like I said, we were really excited about this concept of we had someone in the plant protection clinic for 40 plus years prior to Katie coming in full time as the director. And it's exciting to kind of have a fresh start to look at our relationship and how we can kind of connect on trends and be useful resources to each other. And of course, Katie teaches our plant diagnostics class to the interns, and we're always excited to learn and continue learning from Katie with our most important master gardeners, which is all of you who are here answering questions. And I just want to say that and underscore, thank you all so much for being part of the hotline. We value you so much, and I hope you know how grateful we are to you to slog to URI every week in rain and sunshine and hopefully no snow during the season. And so I will pass it over to Katie Urria-Morwicki, our fabulous plant diagnostic lab director.
Thank you very much, and thank you for having me and for organizing this. This is great. So again, thank you to all of you. Without you, the lab wouldn't function because, as you know, we are a few people and working for operative extension and mainly agriculture, and the lab is me. I get the support from Lisa, from David, but and few students, but you guys are awesome. Thank you for all what you do for us. And as I suggest to Vanessa in the past, it will be good to kind of put a face in the email so you can see me. And some of you, Judith, hello again. Great to see you. Congratulations. I'm so happy to see you here. By the way, she thought that plant pathology is kind of difficult. Uh, she was in the class. And hopefully I don't know, it's up to everybody, but I have a brief presentation. It's kind of a review of plant pathology and mainly the diagnostic process. So I don't know if we want an informal presentation or a conversation, and I just questions and answers. So I don't know. It's up to everybody how you want this to be. And again, the presentation is, it looks long, but it has kind of the main steps that I follow. I have something about the lab, so we can start from there. Probably talk a little bit about the lab and how we function and who are part of the lab, and then we go from there. If I go through the whole presentation, or then we can just start the discussion. They can ask me questions as we go, and we go as, I mean, we have 1 hour, so we see how far we go. And then the main thing, I want to answer all the questions that they might have for me, because this is kind of the space that we have for that.
Great. And if you have any questions that come up during the presentation, go ahead and put them in the chat, and then can have time to unmute and have a conversation after. So I think that sounds good. Thanks. Go ahead, Katie.
Okay, so let me share. And I always have problems with this, so hopefully this works. I am okay. So do you see my screen?
Yes, looks good.
Okay, so again, my name is pronounced, just in case, letter K and letter D. The spelling is kind of long, and I know my last name is long too, but it is Urea Morawiki. And so as Vanessa mentioned before, I took over the diagnostic lab, which it was called Plant Protection Clinic in the past. And due to all the upgrades and what the work that we do, we changed to diagnostic lab. And I am who runs the lab and also diagnostician. So I am the one who diagnosed. I'm lucky to have a student. So I'm having a sales fellow student. She's doing her fellowship in my lab. She started just yesterday. And also I have another student who is graduating. So she's leaving the lab. So she's been with me like a year. Time flies. And she's been learning a lot about diagnostics and been great support, but the lab is that. So just a brief introduction so you have an idea what the lab is. So the lab is part of a bigger organization. We don't function as a URI diagnostic lab alone. We are a part of the National Plant Diagnostic Network. So it's a network of labs around the country. So almost every state and every land-grant university is hosting a diagnostic lab. So the idea is that, the mission of the labs is that we provide quality diagnostics of plant pests and pathogens to our states. So that's why and it's something that you might be getting questions. We are so close to Connecticut, we are so close to Massachusetts, and we always advise customers to bring their samples to their own state. So the plant diagnostic, if you go to the in our website, which is the MPDN or the lab website, there is a link where you can find the MPDN and you will find this map. And the map is clickable, so you can click, you can click and find if somebody from Connecticut. We accept out of state samples because I have a USDA permit. So I am happy to accept the samples, but as a network and as supporting each other, we are in communication all the time. So we talk almost every once a week, once a month. The Northeast, we are divided in regions. So we meet every month and we talk about all the diseases and all the issues that we see in every state. So that for that reason, we support each other in the way that we advise customers to send their samples to their own labs. So the diagnostic lab, as you know, we are part of the cooperative extension service, and we is a fee-based service that we offer, and we provide diagnosis and recommendations. So customers, when they submit samples to the lab, what they get is recommendation, management, the diagnosis, what the pest or what the problem is, and I provide a report where I send all the recommendations and what they can do to manage the problem. So you all are familiar to our website. If not, I can show the website again. Remember that we have those submission forms, but also we have guidelines. So if you go to the website, you see that how it's organized. We have the submission form, and then we have guidelines. The guidelines is a document where we give recommendations on how to collect the sample, which is very important, based on symptoms that they see and the type of material or the type of plant part that is that have the symptoms. So if you have a lead spot, we have recommendations on how to collect those samples, and you all are doing great job with telling people that. But for the ones that are not familiar with our website, we have those resources there. Also, so depending on the type of samples, just remember we have one form that is for plant samples, well, like plant diseases. We have another form, the second form here on the left, the center one of where I'm pointing with the mouse is the insect form, and we have turf. With turf, remember that we request people to submit photos as well. And of course, you guys are the best resource that we have to advise everybody on how to submit the sample. So I really appreciate all the time that you take into telling people how to submit the sample, where to bring the sample, because I mean, with your help, I can do my job, so I can focus on diagnosing the samples. And also, I tell people, and something that I want to tell you, that in the submission form, there is always room for comments. It's good that people give me additional information because it helps me if they don't submit the photos, which lately I've seen that people submit some photos most of the time. So thank you for encouraging people to submit photos, because it helps me a lot in the diagnostic process. Also with the comments, it helps me a lot to have a background of the problem. So sometimes in the form, they just fill the blanks, or they just click where what are the symptoms, what they spray, what not. But it's also additional information that the forms they provide room for people to do that. And also the results is just the reports. So what we do in the lab when we get a sample, because you guys are there, you tell people how to submit samples, but sometimes we can organize a visit to the lab, so you can visit the lab and see how we diagnose the sample. So when the sample comes to the lab, the first thing we do is microscopy. Is we see symptoms, signs, what we see under the microscope. Then I take the decision from there. Usually with microscopic examination is not enough because symptoms can be so similar for different leaf spots, for example. And sometimes the fungus is not sporulating, it's not producing spores, so I have to move to do different testings. So we have, what we do in the lab is we have microscopy, serological tests, and these tests are ELISA. We don't do ELISA, but we do a similar test that is an antibody test. It's similar to COVID test or to any of the pregnancy tests, let's say that. We do incubation. So as I said, 70-80% of plant diseases are caused by fungi. So fungi, they produce different, if you saw in the recordings, they produce fruiting bodies, we call. It's little cases where the spores are, and spores are very important for mycological identification because every fungus have different type of spores. So we do this technique. And the last resource is culturing. When we cannot figure it out, then we move. And culturing is putting some of this tissue, disease tissue with something or with symptoms on plates. Here is Leia. She is my student, so she's plating because we couldn't find anything, or maybe it's bacteria, or maybe it's something else. We couldn't find anything by doing step 1, 2, and 3, and then she is trying to figure it out by culturing. She put a piece in microbiological media. It's called agar. It's food for the fungus or for the bacteria. And then what the result is an isolation of the pathogen that is the causal agent of the disease. So it takes this step sometimes. That's why some people, we say that between 7 to 10 days, because sometimes a diagnosis can take 2 days, can take 3 days, or can take 2 weeks. So depending on the problem. So when people call, also please explain to them that sometimes we need to follow more steps. And so far, it's been good. One thing that I have to mention here is we are trying to send the report through a lab management system. So it's a software where we enter the reports there, and the system is housed at the University of Kansas. So the email that they get is not from URI. The email that they get is from PDIS, is the acronym, and is from the University of Kansas. So it goes to the spam email or spam folder. So that is something that I need to fix tomorrow, and tell Sejal to put in the website that they need to check their spam folder, because I have been two people actually has told me that they haven't gotten the report. I sent the reports, and I noticed that they might have gone to the spam folder. So that's something that I wanted you to know, that in case somebody call or somebody asks about the report, please email me. Some of you have done that. Send me the emails, forward me the emails, but just if they call, just tell them to check on their spam folders. So a brief introduction of what is a plant disease. You guys are so familiar with this, so I'm not going to spend a lot of time. It's just when is abnormal growth or dysfunction of the plant. So we see here a plant is watercress that is yellow, is stunted, compared to a healthy plant on the right that is just normal plant. So diseases like this, like today, is terrible. It's just perfect for fungi. We have higher temperatures, we have all that tender growth. So if there was inoculum there, they are just reproducing like crazy. So that is not good for the growers and for anyone, gardens, nurseries. So yeah, this is a perfect time for plant pathogens. So remember that the disease happen with three parts come together. So we call this the disease triangle. So we need to have a susceptible host. It could be any plant that is susceptible to a pathogen. We need to have the pathogen there. If we don't have pathogen, we don't have a disease. The pathogen can be fungal, like a fungus, can be a bacteria, can be viruses, can be also nematodes or phytoplasmas. I'll talk about a little bit about them. What is the third part of the triangle? The environment. Usually fungi, they need water in the surface to reproduce. So this environment is perfect. We have susceptible plants out there. We have rain for more than 24 hours or 30 hours. Each pathogen has kind of certain requirements. So when we have these wet and cloudy days, it's perfect for disease. So disease happen when these three parts come together. So the plant gets disease. So we know you are familiar with the type of pathogens that cause diseases. So we have fungi, we have oomycetes, and oomycetes are not fungi, they belong to a different kingdom. So let's call them water molds, like Phytophthora or Pythium. We have bacteria. So fungi cause, I think, 80% of plant diseases. So it's the main pathogen that cause plant diseases in the living factors. We have on the left all the living factors, viruses, nematodes, parasitic higher plants, which are plant pathogens too. And then we have other mammals and arthropods that cause plant problems as well. In the non-living or abiotic factors, which is the second main group of plant problems or plant health problems, remember when people water too much or when there is too little, when we have drought, that cause also problems to plants. Lower temperatures. Now I just got an email of cold injury on tomatoes because you see these temperatures at this time of the year are unseasonal. So we are going to start seeing all these cold injury now in the spring. We already have seen a lot of problems with evergreens with cold injury due to drought in the fall and then too much water in the winter and then desiccation from the winds. So chemical injury is also a factor, an abiotic factor, pollution and poor growing conditions. So some of these are caused by us, by humans. Some of the plant problems are we don't have a good planting location or we plant too shallow or too deep. So some of these causes of plant problems are caused by us. So I'm not going to repeat this again. We already are familiar with the type of pathogens that cause plant diseases. So these are the biotic, these are biotic that cause plant problems, insects. Lisa is helping me with diagnosing the insects as well, and herbivores. We know that voles can just kill plants and birds and other biotic factors causing problems. So this is kind of as a in a nutshell, most plant pathogens are fungi or myces that cause almost 80% of the diseases. More fungi, bacteria, and nematodes, they need wet conditions. So the spring is the perfect time for plant pathogens. Phytoplasmas are bacteria. and they need also insects or other vectors to cause diseases, and they are primary vectored by insects. So remember that viruses, and you're familiar with this, that viruses can look like abiotic diseases. So it's tricky to diagnose viruses unless you have the test, the serological test or the molecular test to detect virus. They are very difficult to diagnose. They are systemic, so they are very difficult. And nematodes can cause foliar diseases too. We are familiar with peach leaf disease, right? So that is caused by a nematode. So they can cause also, besides what you have seen, and have I have questions about nematodes. They can be in the soil, but also they can cause foliar diseases. So what is the diagnostic process and how we approach diagnosis? So the diagnosis is a process used for identification of plant problems. In this case, I'm talking about plant because that is what the lab does. So it's almost like you are an investigator. It's a CSI for plants. When you get a call or when you get a picture, that is what you do, right? You start investigating what is wrong with that plant, and then you get all the tools that you can to be able to help your customers and answer the questions of what is this. So what are my goals as a diagnostician? It's a systematic process. I have to see all the clues. So I need to be able to identify the plant problem and the causal agent. That is my main goal, to provide a diagnosis and provide management recommendations to our customers. So be able to narrow down to a few possibilities. And there is when I want to stress out that symptoms can be deceiving. You can see a symptom and you say, oh, that is a leaf spot. The leaf spot can be bacterial, can be caused by bacteria, can be caused by fungi, can be also be chemical injury. So by diagnosing based on photos is really difficult. I get photos and I am septic most of the time. I cannot be sure because I am used to see everything in the microscope or testing them. So I know that it can be some of the symptoms. Sometimes I have problems to differentiate between one or the other pathogens causing diseases by photos. And I have some examples that I want you to see and help you to give more clues on what you can find the answers for. And again, the goal for all of us is to get recommendations for the disease management. So the tools, this is, let's say you do this in the field, in the lab. I have the microscopes and all the tests, but magnifying glass are the best, tool to diagnose problems in the field. And also plastic bags when I collect the samples. When you tell people how to collect the samples, I do have some examples of sample collection, and of course all the tools that they use in the garden. So how to approach a plant problem as a detective? So I carefully examine the plan, and that is what they do. If you get a call, they should go and check their plans. Based on that, they can give you more information. And the submission form has all this information that I asked for. And when a customer calls, they give you all this information. What is happening, when happened, do you spray anything, how do you water your plants? So all these questions really help to figure out the problem. Looks for clues. They might be abiotic. They might have sprayed something. They might have pruned the plant, and maybe we have low temperatures. All this information is very important because it gives you an idea of what the problem could be. So steps, what are the steps that I follow when I get a sample? So, and you do the same. what is the plant? What kind of plant? Some of what? For example, we got a sample today, and the sample in the form they put that it was a boxwood. When we got the sample, I noticed that the leaves look different than a boxwood. It was a holly. So the first thing I do when I get a sample to the lab, because usually people buy houses, they don't plant these plants. They have a landscape and they have gardens and they don't know what they get. So in this case, it's very important for me to know and confirm when I get the sample that I get the right host. So in this case, it changed the whole thing, because the diseases that are common to boxwoods are not the same that are common for hollies. So they have different sets of diseases. So knowing the host is very important, because that is kind of where I start with my diagnosis. So describe the symptoms and signs. It's very important, because then I'll go through this, but I'm gonna give a brief talk here. Look for patterns, determine the timing of the symptoms, ask questions about the plant history, and also now and you also, you use mostly Google, and then it's very important there that you put extension or edu, because those are the most reliable, and you all do this all the time. So again, we come back to what is normal in the plan. Once you ID the plan and you have the right host, what is normal? In this case, we have this white pine. The inner needles are yellowing. This is a clue that is probably natural senescence. It's something that happens every fall. So that is something that you know, and kind of looking at the symptoms is something that you have to think, okay, in this time of the year, what this plant do? So then if it's the fall, if it's in the fall, it's natural senescence. If it's in the spring and the symptoms are in the new needles, that is probably a disease. So you have to think about all this. We have some ornamental plants that are variegated, and sometimes it can be confused with viruses. This is are kind of so obvious. This is just examples that I have, but there are others that are more difficult. So sometimes people is not familiar with what is normal in the plant. So for you, it's always important to put what is normal is a hybrid, is a variegated cultivar. Those spots are normal or not. Describe the symptoms and signs. What are symptoms? So symptoms are expression of the plant. So those symptoms, they have names. In the left top part, we have chlorosis. Chlorosis is when we have this yellowing. Sometimes we have browning, and browning is when the plant looks all brown or blight. We have blight as well. This is leaf spots, and leaf spots can be caused by different pathogens, not only fungi. They can be caused by bacteria. That's why it's very difficult to diagnose them by photos, because for this you have to use the microscope, you have to do mount to be able to see the spores or to be able to see streaming, and then when it's bacteria you have to do further testing. Blotch. So these blotches are different than leaf spots because the edges are kind of blurry compared to a leaf spot. We have cankers. You guys are familiar with cankers. Wilting. You're familiar with this symptom. This can be caused by also bacteria or fungi. And in tomatoes, it's really difficult to diagnose the vascular pathogens. By the way, we have a really bad pathogen that causes wilting in tomato. It's called oh my gosh, it was here and now is Ralstonia. It's called Ralstonia solanacearum, and it's a very bad pathogen. So I am on the look for that pathogen because it was diagnosed uh, in a sample that didn't come to my lab, and it was a sample from Rhode Island. So if you get any calls about wilting on tomatoes, please advise them to submit a sample to the diagnostic lab because those pathogens are soil-borne pathogens and are really bad pathogens. So I have tests for most of these wilt pathogens, and also I have tests the problem with soil-borne pathogens, as you know, they survive in the soil. So detection is very important. Early detection is very important, and it's very important to know if the nature of the pathogen is bacteria, because if it's bacteria, next year they can treat the soil, or they can do something with the seed and plant a resistant cultivar in that soil. So for that reason, testing is very important because we can kind of ID the pathogen, so then management is easier for growers and also for homeowners and for everybody. Root rot pathogens. Root rot pathogens, sometimes you see wilting of the plant or dieback, or you see the symptoms of root rot pathogens. You don't see the symptoms until you pull the roots and you watch the roots and you kind of clean the roots. That is when you see the problem is a root rot pathogen. But sometimes the symptom, it can be browning, it can be yellowing, it can be many symptoms. And I have some pictures later. So signs, and this is where my diagnostic uh, tools are the best to diagnose for. I always look for signs because the symptoms are so difficult. So when I get something in the lab, what I see, I look for pycnidia, or I look for different fruiting bodies. The fruiting bodies have different names, so we are not going to describe all the names here. But I look also for fungal structures, mass spores, mycelium, or pycnidia. Or I look for streaming. When I do mounts in the microscope, I look for this. So this is, and as you know, sometimes you're so familiar with some of these diseases because every year you get questions about powdery mildew, right? And with powdery mildew, sometimes it's easier to diagnose than any other disease, because if you look a good picture, if the customer say, oh, I have roses, or I have peonies, or I have whatever, everything gets powdery mildew, or cucumbers, or pumpkins, and that is one of the easiest disease to diagnose because of the signs, because you actually see the pathogen. So this is another one that, for this, I'm never sure what it could be, because it could be Cercospora, it could be something else, it could be Phylosticta. Phylosticta produce this fruiting body. So when I get this, and I also need to incubate, I need to do further tests, my microscopic tests. This is something that I wanted to point out. When we see in boxwood, we see these symptoms, and sometimes you get these pictures, right? When we see, they call blight, we see this browning, and it can be in one or two boxwoods. And then there are different things that can be happening with this boxwood. It can be Volutella blight, it can be macrophoma leaf spot, or it can be really a bad disease that is called boxwood blight. And you all are familiar with boxwood blight because you have seen samples of boxwood blight. So even for me, when I get a sample of boxwood, I need to incubate them, because by the symptoms, I cannot know what is going on. So I need to incubate, and for that I need to use the microscope to be able to differentiate between these two opportunistic pathogens that they attack only weakened plants, and then ID them. But for boxwood blight is even more challenging. I need to incubate almost for 7 days and then to look at the type of spores, because by the symptoms, it's just really difficult, and it's a disease that once it established is really hard to control. You have to use a lot of pesticides. So even for this, it's very important that if they submit samples or you get questions like this, it's very important that they submit samples. So look for patterns. So, what part of the plan is affected? You guys are really good in telling people on what to submit, and I have a photo later of a submission that I got, and I got the whole plan. Always the whole plan is the best sample for me, but of course, it cannot be done all the time. It can be done for small plants, maybe a little tomatoes, some of the flower beds, but some of the times it cannot be the whole plant. But look for patterns. It happens to the inner leaves. Are the symptoms the same? Look here in this tomato. If the symptoms are the same in every leaves, or the symptoms are random, or here in the landscape we see that all these boxwood, they look the same. This is usually a random pattern, as I have here in the left side. It is probably a biotic is caused by a pathogen, whereas where a no random pattern, so an uniform pattern, might be just cold injury or winter injury. So if we see that the pattern is random, it's probably a plant pathogen. If the symptoms are not random and all the plants look the same, it's maybe a abiotic factor. So can be, you have to ask the question, happen to one plant, happens to all the plants in the same area? That is very important. This is a photo of our famous winter injury problem that we have this year. As you see, it was happening to all these laurels. When is the time when that happened? It's important to know if the symptoms develop suddenly in a week, or the symptom has been progressing. When did you see the symptoms starting? Like 2 months ago, and it's been progressing, getting worse, or it happened overnight, as some people tell me. Oh, this just is sudden. So usually gradual development is more caused by the disease, a pathogen, and sudden symptoms usually is called injury or environmental factor. So something like this. And depending on when they saw the symptoms, sometimes you see this with a soil-borne pathogen, but you start seeing the symptoms developing gradually. They don't show up once or overnight, as people say. So this is the rhododendrons that we have the problem this year. This was suddenly. So we just saw it from 1 week to another. And this is what I sent the email about. This rhododendron already had the Botryopsphera canker. And this is all injury, and this was doing really bad. We tried to prune, we tried to do something, but with the cold or the drought in the fall, and then with the winter injury, it died. So, but it already had the disease, and I saw the cankers, but that wasn't the cause of the disease. That was kind of a secondary factor. So ask question about the plant history, and you know about that. I'm not going to spend a lot of time. This is in the form, and you have seen it. So, and you always ask. This is very important of having clues of what's going on, when they water, windy location. This is what happened this year. The plants where the wind was coming stronger is where we saw more cold injury. This is poor growing conditions, equipment damage, and then you go to the books, in my case, and then you use extension or EDU in all your Google search. And also I will recommend use the universities in the Northeast because we get similar pathogens, so that will be good. And this, I will just not going to repeat all of this. So, good sample versus bad sample. And this was something that happened with a sample that we submitted recently. So the homeowner has some beds where nothing was growing. And the first sample that he submitted was only soil. I didn't have any plants. So when I got the sample, I said, oh my gosh. So I have to call him and I told the lab cannot diagnose plant pathogens in soil because soil has so many pathogens, beneficial and pathogenic ones. So in the lab, we only diagnose from the plants. So I called him and he was just so happy to get my call and he submitted a second sample. And this is his resubmission. I also told him to wrap the upper part of the plant in another bag, to put the soil in a bag and then tied. He tied this so the soil wasn't in contact with the upper part of the plant. That is really important for my diagnosis because sometimes I do tape mount or microscopic examination of the leaves, which I cannot do it if there is soil all over the leaves. So it's very important to advise people to separate with two different bags to put the tops and cover them, or to just put tie this with a different rubber band or something so the soil is not in contact with the plant. And this is what he did next time. The first sample, I didn't take a picture, but the plant was in one bag with the soil, so the soil was all over the top of the plant. So this is very important, and this is a perfect sample because then I can do my diagnosis. Another good sample, I got this sample that is a holly. They wrap, they provide the whole plant. And in this case, it was winter injury. I tested the roots. Nothing was positive for soil-borne. I asked them if they wanted the plant back because I took the sample I needed and they didn't want it because they say it was just so awful. So they just have more plants. And so this is a perfect sample. It's the whole sample. And I was able to test, and it was a winter injury. This is also a good example of a good sample. You see how they put the soil in a plastic bag and the roots. So this soil was good sample because it has the roots there. And you see how they put everything in plastic bags. They even put the submission form in a Ziploc bag. So this is very important because for me, that is the dream sample when I open a box and I see that everything is separated and organized. So this is an example of a good sample. And they got probably good advice for all of you because I was like, oh, this is perfect. So this is an example, and I just probably I'm going to stop because I don't have a lot here. It's just one example of insects. So I just wanna answer your questions if that is okay.
Great. Thank you so much, Katie. There were some questions in the chat. Do you want me to narrate or do you want to take a look and kind of go through?
So if you don't mind to read the question, and then I can give you an answer.
Cool, cool. So Vivian is wondering if it's possible to email the hotline back when the diagnosis was sent through the hotline to the lab.
And that is a question that I have been getting as well in the past. And the problem is that we have a client confidentiality, and I cannot share the diagnosis from specific samples because that is client confidentiality. So what I can do is that I can provide, like, if they still are wondering about samples and they have questions, they can email me. And in general, I can tell you what I seen with the samples, but I cannot provide a specific diagnosis because those are private for the customers that they submit to the lab, unfortunately.
Okay, another question. Thank you for clarifying that. UMass has a monthly newsletter that includes pests and diseases prevalent in their state. He says, I know you sent us a response for the winter dieback. Would you have capacity at the lab to send us similar information when you are seeing prevalent diagnoses?
Sure, sure. I just advise probably the contact now that we have Jonathan. That's really helpful because I don't usually I get so busy. I am wearing a lot of hats, so I get super busy in the season. So having somebody who maybe can visit me often and say, "Oh, what's going on?" Or I mean, he can come to the lab anytime, and I can tell him, "Oh, you know this, I seen this," and you see the patterns because that is, I think, what Jonathan is will do. Keep us connected, and I will give him a heads up what is going on. So yeah, we work on that because yeah, we haven't done that. So yeah.
That would be great. And then if day leaders are seeing, well, if anyone is seeing, prevalence of a lot of similar samples, you want people to send that over to you as well. You said if we got 3 or more of similar things, so they can either send that through Jonathan or directly to you. I don't know if you'd rather have it go through Jonathan now that we have him. Let me know.
Okay, yeah, that I just heads up. We might have some problems in a week or two because of the rain. So you are going to be get busy with calls because this is just yeah.
I remember listening to Larry Englander and he'd get really excited. So get excited. You're going to get to see some interesting fungi coming. What are you seeing in ornamental damage as a result of climate change? For example, some of the spruces which are native to alpine regions are failing. And Alan says Christmas tree farms have shared their concerns with him in the past couple of years.
Yeah, I visited a Christmas tree farm and uh, I found a disease that I never, I haven't seen and it's not seen often. And is called, I'm double checking the name, those pathogens, as you know, is sudden spruce needle drop and it's a fungus. And I'm looking at because I never seen it, and I'm looking for the right name. I can put in the chat. I'm going to put the name of the disease, and that's something. So we are going to see more pathogens that we haven't seen, and that's something that I told Vanessa because the weather is becoming more erratic, so storms are going to see more often and worse than probably we are used to, because that is what climate change will cause, right? We will have more rain when we didn't have that much rain, storms, and I'm just typing. I'm sorry, I get distracted and so this is something that I haven't seen. Sudden sudden needle drop in spruce. And I just saw I diagnosed last week. And this is the name of the pathogen is I didn't even CTO. And it caused needle drop. And I was lucky that this was like early stage. That is the name of the pathogen. And I found it's been reported in Massachusetts, in Illinois. So and also I think was, yeah, Illinois, and I found different ones. But I haven't seen, I need to ask the diagnostician in the Northeast if they have seen it. Also, I seen a disease that I haven't seen before in Japanese umbrella pines. I'm still working on that. It's a fungal disease. So if you get a call, please tell them to submit a sample because it's a disease. Japanese umbrella pines are not pines, as you know, and they are very resistant to diseases until last year. And then what the symptom is that you see that one branch start dying, and then another branch the next year, and then it's like that. I have landscaper and arborists. They have provided me samples, and I still working in the ID of that fungal pathogen. It kills the twig. It kills the whole. It causes like a. It's not canker because it doesn't cause the crack, but I see the fruiting bodies there. It produced pycnidia. So it's something that I is still working on this. I don't have an ID yet, but it's something that we didn't see before.
All the fun new pathogens. There was a question about what we should do when you see someone with spotted lanternfly nymphs or any other life stages. Are we sending them to DEM or what should we do?
Just kill them. I'm sorry to be this hard, but yeah, you just remove the eggs or the nymphs, or if you see an adult, just squash them. Because yeah, you can report to the DM because now it's everywhere. So it's kind of reporting, but take action too. They are spraying also for spotted lanternfly, and Lisa can be a better resource than me because she does this outreach all the time. But as far as I know, we don't need to report it anymore because it's everywhere in Rhode Island already.
Great. We'll get one of those trainings scheduled with Lisa to get a little bit more, but
Yes.
as far as you know, don't need to. No
No,
need to
because it's
report.
everywhere. Yeah.
Ugh.
But remove it, and Lisa can update you with the latest. Probably you can ask them and then send an email to everybody just if I am no correct.
Okay, will do. If anyone wants to unmute and ask a question, feel free.
It's Paula. I wanted to go back to the issue around sharing email responses to regarding plant diagnostics that sort of came through the hotline. And you mentioned, the issue is client confidentiality. If we tell the client, to put on the form that this information can be shared with the hotline, would that satisfy your issues around confidentiality?
No, because we have a lab management system, and since we are a network of labs, we get funding from the plant diagnostic network, and this is part of the agreement that we provide that diagnosis, and it's part of, even if the customer wants to call you back, and you can tell them, oh, can you call me back because I want to know, that's up to them. But from me, I cannot provide that information because all the diagnoses are uploaded to the repository, and it's just is more like, and now I am applying for accreditation. So it's just that is the problem with is the whole network kind of thing.
Understood. Thank
So,
you.
but yeah, but you can just tell the customer. I mean, can you tell me what it's up to the customer as well.
I think it would be a lot more professional on our side to just wait for Jonathan and Katie to have that back and forth and see what the trends are, rather than being like, hey, on the side, let us know what it was, even though you shouldn't really tell it. I don't know. That feels a little like we shouldn't be doing that. So let's see how this works out with the back and forth communication. Go ahead, Sally.
When we send our clients information or respond to them, we usually send them written fact sheets. Sometimes we will send them saying, this may help you find, understand what's happening with your plants. Do you do anything like that when you respond to customers? Do you send them information, written information about it?
Yes, all the time.
Oh, you do?
Yeah,
Okay,
yes, I do.
good.
So that is part of the diagnostic report, and I don't have a diagnostic report here. But next time that we do this, I can provide what the diagnostic report is. And yeah, I use fact sheets, and I kind of put the relevant information for the management in different bullet points. I say, this is the pathogen, this is the common name, these are the recommendations, and then I send a link of the fact sheet that I think is the most appropriate, one or two if I need to. But yeah, I use them. They are perfect. Yeah, so yeah.
Very good. Thank you.
Like, and you all know my email, right? So just if you have any questions or anything, I try to be very good in answering questions. Sometimes it takes me a while a day. If I'm busy, if I am in the field, it takes me some days, but I'll try to be responsive on my email. So if you have any questions, I am here to help you. And Jonathan and Vanessa, thank you very much to all of you. And thank you for volunteering. Really, this is just amazing. I've been working in another state, and we didn't have this support either. So yeah, I'm really happy to have this support here in Rhode Island, and thank you for all you do for us and for the community, because really this helps a lot the community and everybody. So thank you.
One more thing. It would be nice to know how many of the clients we refer to you actually follow through and send you samples. Is there any way we could know maybe how many clients actually reached you or went to you in a certain day or week or something? How many referrals you got from Hotline that worked out?
That is just I don't know. That is, I don't think we can just yeah, it's just
Yeah.
hard because
One of the notes I put in was if there was something on the form, a question that said, where did you hear about us? Then you could have data collected in terms of hotline, how many referrals came from the hotline.
yeah, I don't know if that is our information that should come to the lab. I'm so sorry. I don't know. But Vanessa, Jonathan, and I will try to figure out a way to do that for sure. That's something that, because again, is client confidentiality, so I cannot disclose who submits, put a list of names in a list. Because when they submit a sample, that is kind of an agreement, a contract of confidentiality. So I can put maybe a host, like what kind of plant was submitted, and maybe if you, I mean, we can talk about that with Vanessa, but I cannot provide a list of names of people that submit that. But we can, yeah, Alan, we will take your suggestion into consideration and see how we can, but I don't know. I will say that most of the plant samples are submitted because of your suggestions. That is what I can say. That probably most of them are, because I can see how, when they fill the information, because when they good, and some of them they tell me that for example, when they say in an email, they say, oh, I talked with the home hotline and they told me this, the master gardeners told me. So we can have a trend, or maybe, I don't know how we can, but we will talk with Vanessa and Jonathan and see. But I would say that it's so effective that probably most of the ones that you suggest to send a sample, they do send a sample to us based on what I
Thank you.
have heard.
How
So
often
it's
are you
very
able to get
effective.
out? How often are you able to get out in the field? Like there's a lot of times people will say, a lot of times we make a referral to Rhode Island Nursery Landscape Association for an arborist to go out to examine something. And I just didn't know if that's something you do on a regular basis or a sporadic basis.
We do, I do that, and there is a fee, so they can contact me if they want me, like homeowners mostly. If a homeowner, I've been there, and there is a fee for my visit and for collecting the sample. But yeah, I do that service when time permits me, when I am not so busy. But yeah, if there is a homeowner that wants me to visit and assess the problem, I'll do that. So you just have to email me and ask me, or the homeowner to email me. We can do both ways. They can email me and request a site visit, it's called. So yeah, I'll do that if they want me to. And sometimes that is so good because when I go and see the problem, it's just so different than when, for example, they send a picture.
I didn't know that. Someone asked Mamie that the other day and she said, I don't know. I was wondering the
Yeah,
same
they
thing.
can. Yeah, if I have time, I try to make time for that as well. Because I like to go to farms. I like to visit homeowners. The problem is the time. Again, I want to thank you because people, I see the difference because people is sending me a lot of photos and they help a lot. And with the TARF samples, everybody sends photos. So thank you. Yeah, that is because of you.
Thanks, Katie. And I put together a list of takeaways. I'm going to send it over your way in case I missed anything. And then I'll share it with everyone. But I put it in our shared drive under Hotline trainings. And I will share that out along with the recording because I know there's some folks who aren't here that will want to watch this later. Cool. Well, it's nice to see you all. And hopefully we can do our next training in person and hang out. But stay dry, everyone. Don't go anywhere if you don't have to. Thank you, Katie.
Thank you very much for having me. And yeah, I'll be in touch. And good luck in the season. I hope to see you
Yes.
soon.
Thank you.
Stay strong. Have a good one. Bye.